defenses, that's going to be things like structural, mechanical, and biochemical. They prevent the microbe from entering our body. However, if the microbe is able to overcome number one, then number two should be the next form of line of defense, which is going to be the innate immune system, which will attack the invader, the foreign body, the microbe, or the virus, and hopefully eliminate it. However, if number two is overwhelmed, then our third line of defense will be the adaptive immune system, and then this is going to be activated. You should definitely know this slide, and that they occur in this order. So lines of defense now shown to you at like the actual level of the tissue. We have the barriers, non-specific surface defenses, and things like sweat, sebum, things of that nature. Then going a little bit lower, you're going to have the innate, which would be like your non-specific immune defenses, complement system, things like that. And then finally, adaptive. If this one is also overcome, the body will learn how to strategically make a response that should be most efficient. So our immune system consists of two parts. You need to know this. Innate and adaptive. Okay, we're not talking about the structural defenses. Now we're talking about the actual immune system, innate and adaptive. They differ in the ways that they recognize the microbes. That's important. They do use the same means to destroy the microbes. So they have different ways of identification, but then they ultimately kind of converge together and will respond in their destruction relatively similar, identical. They have evolved to work together, but the innate system evolved first. Animals excluding vertebrates are going to lack the adaptive immune system, and they must slowly rely on their structural defenses and then the innate. They don't have the adaptive. I took this picture from another textbook. I like the comparing and contrasting tables. So, characteristics, speed of the response, the innate immunity is very rapid. It begins to act just within a few hours of infection, versus adaptive is very slow. It can take days or weeks to act. Distribution in nature, innate immunity is going to be present in all animals. Adaptive immunity is going to only be found in vertebrates. Variation over time. The innate immunity doesn't really change. It's going to it's got the same mechanisms of defense. However, the adaptive immunity, thus the name implies, it is adaptive. It will change, it will evolve, those types of things. Mechanism for recognizing the pathogen, that is going to be a little bit different. Innate immunity recognizes classes of molecules shared by a group of microorganisms. Adaptive immunity is going to recognize unique molecules on a particular pathogen. Okay, we'll be talking about that. Number of molecules recognized. Innate really only recognizes a few, and then adaptive is like infinite. Like we don't know. We don't know if there's like a point where it stops or anything like that. It doesn't recognize anything. It's fully saturated. And doesn't change during the response. Innate immunity really doesn't. It's pretty consistent. Adaptive will improve, and then his response will get faster. It's very, very strategic. So a summary of immune system defenses. This is a good slide. You should know this. Immune system, the body's defenses. Here we're talking about innate, which is going to be non-specific. It's very generalized. We have surface, and then you're going to have cellular, chemical, and coordinated. At the surface, that's going to be things like your skin, mucosal membranes. They're going to contain macrophages there that will also respond. At the cellular, chemical level, we also have normal flora that is going to respond, things when you get inside the body. Then on the other spectrum, we have adaptive, which is a highly specific form of immunity. That's also going to be divided into two subcategories. You have cellular, you have humoral, which basically means like liquid, and we're going to talk about fluid. We're going to talk about adaptive. That'll be the next lecture. Innate immunity, the characteristic. This is a host defense mechanism. It's very rapid. Again, this is basically taking the information from the previous slide and just rewording it for you. It's broad. It does not adapt to a particular pathogen, but recognizes traits common to multiple different types of pathogens. Are some people missing this slide? Is that when you're really flipping through it? Yeah, I updated the lecture on Moodle. This was one of the tables that my friend added in, which I thought was helpful, so it's up there. You guys have it. It's on Moodle. So it's not mediated necessarily by lymphocytes. However, this becomes highly debatable. The innate immunity doesn't require, it's not mediated by lymphocytes, but when we get going, which I've talked to a few immunologists about this, and they say it all depends on the professor that you take. Whether they're going to require you to know that, like, natural killer cells are in innate or adaptive, it's all different. So she told me to kind of tell you guys whatever I want. And I was like, okay, so we're going to talk about how we're going to classify all of that. But it's one of those things, like I got this Venn diagram that I need to give you guys, and it shows the innate and the adaptive and the things in the middle, and like natural killer cells kind of get lumped in the middle and stuff like that. You know what a Venn diagram is, like the three circles? Okay. Anyways, so it's always present. The innate system exposure. So innate immunity includes these three things. I want to make sure you know this. The slide says test. Complement. First response to invading microbes, a set of microbe-attacking proteins in the blood, lymph, and extracellular fluids. We're going to talk about this. There's a ton, and they're all named boring stuff, like C9, C5a, C3, C3a, and C3b. I'm going to tell you the ones you guys need to know. But it's a little bit intimidating, but when it comes down to it, I think you need to know like 5 of them. Okay, so don't freak out about it. Number 2 is going to be phagocytosis, the first attack on the invaders. The leukocytes will destroy pathogens by phagocytosis. And then number 3, inflammation. Inflammation is going to involve the recruitment of phagocytic leukocytes to the site of the infection, gathering of plasma proteins. You're going to have things like cytokines, which you can end up having the accumulation of pus, redness, heat, which this is all actually good. These are positive things, as long as it's not persistent. Everybody thinks inflammation is bad. Inflammation is good. It lets you know that there's something going on. What's dangerous is chronic or long-term inflammation. You shouldn't be inflamed for a long period of time. If you are, then there's a serious problem. The complement system. So we're going to talk about this. This is the cascading sequence. It's a very complex system of more than 30 different plasma proteins. Okay, you don't have to know 30. They act in a cascading sequence. A cascade is like dominoes, where one does something and the next one reacts and does something. So a cascade of enzymatic cleavages to help eliminate infectious microorganisms. The complement is found in blood, lymph, and extracellular fluids. Proteases are going to be enzymes that cleave proteins. They circulate in an inactive form, but can be activated to break down other proteins into smaller peptides. That's their job. It can be activated by 3 different pathways. We've got three stages of defense. We're going to talk about the 3 different pathways. Our class is really going to focus on the alternative activation, but there are 2 more. We're going to address those, but we're not going to go into any details. And they provide 3 stages of defense: lysis, which would be bursting of the microorganism; attached to the microorganism, which kind of act as like a flagging mechanisms for phagocytosis; and then, of course, recruitment of inflammatory molecules.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2a and C2b, and C4 into C4a and C4b. C2b and C4b combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3a and C3b.
This is where they both got
In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3a and C3b are constantly being formed from C3, but without activation, they are soon destroyed. C3b reacts with the proteins, factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3a and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3a is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3a and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properdin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5a and C5b. C5a enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components, including C6, C7, C8, and C9, to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
Okay, so you don't have to know all of that, but we're going to get into it. There's three complement pathways. There's the alternative, there's the lectin, and then there's the classical. Classical is really focusing on antibodies. I want you to know that, but I want you to know that we're going to talk about alternative, which does not rely on antibodies. Antibodies is going to come up later. So via the alternative pathway, the pathogen surface is going to create a local environment conducive to the complement activation. So it's going to be one of the first to act. When it does, it's going to activate the complement system. And this has got to do with C3. So you do have to know of all those proteins. You've got to know about C3. You've got to know that there's C3 floating around in your body, which isn't floating, but running around in the lymph and the blood and stuff like that. It's going to react with water and split. And when it does, it's going to form fragments. And those fragments are C3b and C3a. Designated with lowercase letters. See this? A. See that's lowercase? That B is a lowercase? That means a fragment. That's why they're written in lowercase letters. So that's definitely something, if you're taking notes, I would have written down, because that's going to be a huge clue. Because if on your exam, if I do something like, I say C3a, lowercase, then you know that means C3b has already been split. So it kind of it's a trick to let you know where you're at in this cascading sequence. So that lowercase letter means it's been a fragment. It's already been split. So once that happens, we're going to talk about what you need to know there. We're going to have a recruitment of the inflammatory cells, opsonization, which opsonization is just a fancy way of saying that your body flags a foreign microbe that shouldn't be there. And then phagocytes, or cells capable of phagocytosis, will identify that flag and engulf and destroy that microbe. However, remember, we have talked about that there are certain microbes that want that to happen. And then we also have different types of proliferations that can occur. And hopefully the primary result will be death of the pathogen. So complement activation always leads to the cleavage of C3. We need to know this. So you've got C3 running around in your body. It's going to be split with water. That's how it's cleaved to form C3a and C3b. C3a is going to be involved in inflammation, which is going to be the recruitment of the inflammatory cells. C3b, lowercase b, binds to the bacterial surface or the pathogen. You can almost think of that little b to represent binding, okay, because it's going to attach to the pathogen, which will flag it for opsonization. So regardless of the pathway, okay, regardless whether it's the alternative pathway, the lectin, or the classical pathway, this is all really going to be the same, which is one of the reasons why I thought this was a really good place to start, because it basically jumps right down to here. Okay, and the alternative pathway is going to be one of the first to act. So that's why I want to make sure you know about the alternative pathway. Here's another slide about it. Complement pathway, focusing on the alternative, even though we know that there are three, and we know that the alternative pathway is lacking antibodies. So, complement activation can occur via classical or lectin pathways. We're going to focus on the alternative pathway. The first thing that's going to happen is hydrolysis of C3. You've got C3 naturally circling around your body. That will sometimes encounter water and cleave. Okay? When it cleaves, if there's bacteria present, then those fragments will bind and they'll do what they're supposed to do. If there is not a pathogen present, then it's just going to degrade, because there's no point in making all of those fragments